In this lesson, we cover a bit more detail about translation. First, let's start by talking about the major players. One that we need to talk about is transfer RNA, or tRNA, and its job is to transfer the correct amino acid into the growing polypeptide. So we know that messenger RNA has been transcribed off of DNA, and now we need to get the three-letter code of codons on the messenger RNA to tell us which amino acid is going to be put into the growing polypeptide. So here I'm drawing this cloverleaf-like structure. The blue is the transfer RNA, and the red is the amino acid that it is going to carry. Now, transfer RNA is made of RNA, and the RNA, although this is all one single strand, actually complementary base pairs with itself in some regions to give it this cloverleaf structure. And then there are going to be three bases at the bottom that are unpaired, and this is the anticodon. And this is the part that's going to bind to the codon on the messenger RNA, and that's how the messenger RNA specifies which amino acid goes next into the growing polypeptide. So if this is CCA and the anticodon, then the codon that it would be binding to on the messenger RNA would be GGU. So now we can see how we get the correct matchup on this end from the codon on the messenger RNA to the anticodon on the tRNA by complementary base pairing. But what about at this end? Because getting it right at this end is equally important. Well, that's the job of tRNA activating enzymes, also called aminoacyl tRNA synthetases. And they're enzymes that join the correct amino acid to the correct transfer RNA. So they put the amino acid on the top of the tRNA. And each enzyme binds one type of amino acid. For example, there's an enzyme for putting phenylalanine on transfer RNA. There's an enzyme for putting proline on transfer RNA. They each bind one type of amino acid, but they'll bind all of the transfer RNAs that recognize codons for that amino acid. So for example, because of the degeneracy of the code, there's more than one codon that will call for phenylalanine, and the tRNA activating enzyme that goes with phenylalanine will bind all of those. So there are 20 different tRNA activating enzymes, one for each amino acid. Now, the tRNAs don't actually look like this in 3 dimensions. They look more like an L shape or a triangle, so I'm going to draw it like that with the amino acid here, and then you can imagine that you've got the tRNA activating enzyme that has specificity, enzyme substrate specificity for both the amino acid and the tRNA, and partly that has to do with it matching up with the anticodon. So tRNA activating enzymes put the right amino acids onto the right transfer RNAs. Now, ribosomes are the complexes, the machinery, that do the translation. They are complexes made up of proteins and ribosomal RNA, which gets called little r RNA, so RNA like that. And they're made up of 2 subunits. There's a large subunit and a small subunit. And the large subunit has 3 different sites on it. The A site, the P site, and the E site. The A site is where the incoming tRNA carrying a new amino acid comes in. A is for amino acid. P is where the growing polypeptide chain is made. P for polypeptide. And then E is where the transfer RNA that has given up its amino acid exits. So you can imagine transfer RNAs coming in this way and leaving this way. And the messenger RNA lies down here in between the large and the small subunit. The polypeptide is going to come snaking out of the top here. There's a channel through the large subunit of the ribosome, and it's going to come snaking out the top here and fold as it comes out. So that's the structure of the ribosome. Now let's look at the process of translation. So this is a diagram that we've seen before in a previous lesson from Campbell Biology showing the process of translation with transfer RNAs here shown in the light blue bringing amino acids into the ribosome. We see the messenger RNA in red, and we see the complementary base pairing between the anticodon on the transfer RNA and the codons on the messenger RNA. Notice that the messenger RNA is moving through this way, so you can think of that instead as the messenger RNA staying in place and the ribosome moving along the messenger RNA in a 5' to 3' direction. Again, ribosomes move in a 5' to 3' direction along the messenger RNA that they're translating. Notice also that you can see the A site with the new transfer RNA with an amino acid coming in, the P site where the polypeptide is being formed, and the E site where the transfer RNA without its amino acid is exiting. Before translation starts, the large subunit and the small subunit of the ribosome actually aren't together. Translation starts with the small subunit and the first transfer RNA, which is always carrying methionine, and that has been called for by the codon on the messenger RNA, AUG. The small subunit, the first transfer RNA, and the messenger RNA join together. So you've got the small subunit, you've got the messenger RNA, and then you've got the first transfer RNA, like that. They join together, and only after that does the large ribosomal subunit join. So methionine is the first amino acid in every polypeptide, although it can get cut out later in post-translational processing. After that, successive transfer RNAs are going to come in carrying their amino acid, complementary base pairing happens here, and then a peptide bond is formed between the growing polypeptide and the new amino acid. The whole ribosome then moves along 3 bases or 1 codon at a time, and then you get the next amino acid. And the bond formed between those amino acids is a peptide bond. And this process keeps happening, keeps repeating, until we encounter a stop codon on the messenger RNA. And when the stop codon is encountered, instead of bringing in a transfer RNA, stop codons call for the binding of a release factor. And a release factor, instead of bringing an amino acid, triggers the addition of water. Water hydrolyzes the bond between the transfer RNA and the growing polypeptide chain, so this breaks off here, and now the polypeptide is free and folds, and there can be post-translational processing that happens as well. So that's an outline of the process of translation. Now let's finish up by talking about the 2 different classes of ribosomes, free and bound ribosomes. Free means that they are free in the cytosol, and they synthesize proteins that are going to stay in the cytosol. So they're free, they're just floating around, they're not bound to membranes. Membrane-bound ribosomes, or bound ribosomes, are on the rough endoplasmic reticulum. That's what that abbreviation stands for, and they make proteins for the endomembrane system. The endomembrane system includes all the membrane-bound organelles like it includes the nuclear membrane, it includes the rough ER itself, it includes the plasma membrane, so all of those have proteins embedded in them, so those proteins would be made on bound ribosomes, also for lysosomes or vacuoles. So any proteins that are bound for either the lumen or the membranes of things like that, they're made on bound ribosomes. Also, proteins that are going to be secreted out of the cell are made on bound ribosomes, because then they get into the Golgi and then they are processed for export out of the cell. Now, interestingly, bound ribosomes actually start out free. They start out free in the cytosol, but a sequence on the growing polypeptide, so here's our ribosome with our messenger RNA, here's the polypeptide snaking out of the top, there's actually a sequence here on the growing polypeptide called a signal sequence that tells the cell that this is meant to be targeted for the endomembrane system or for secretion, and it actually halts translation until the ribosome can be moved onto the rough endoplasmic reticulum. So bound ribosomes actually start out free, but a signal sequence here in the growing polypeptide arrests translation until the whole thing can be moved to the rough endoplasmic reticulum. So they start free, and then a sequence on the growing polypeptide itself actually targets the ribosome to the endoplasmic reticulum. So that is an overview of translation.